Imagine you're walking through a damp autumn forest.
Okay, setting the scene. I like it.
Yeah, and you reach down and you pluck two perfectly identical, beautifully strange mushrooms right out of the leaf litter.
Just totally identical to the naked eye.
Exactly. They both have these stunning metallic silvery purple caps. They feel exactly the same, like a dry, satiny texture against your thumb.
Right.
And even if you put their spores under a microscope, the tiny rust-colored warts on those spores match perfectly.
They're a flawless visual match.
Flawless. But here's the wild part. Genetically, if you look at their DNA, they are as distantly related to each other as, like, a human being is to a kangaroo.
It is the ultimate biological paradox, honestly.
It totally is. And welcome to the staggering taxonomic nightmare of the Cortinarius genus.
It really is a nightmare. Because we've spent, centuries trusting our vision to categorize the natural world.
Right, assuming if it looks like a duck and acts like a duck, it's a duck.
Exactly. But fungi, and particularly this specific genus, they're actively proving that our eyes are completely inadequate tools for understanding the forest.
And that is exactly our mission for this deep dive.
Yep.
We are tearing apart the chaotic, impossible world of Cortinarius.
Which is no small task.
No kidding. I like to think of this genus as a massive, sprawling family reunion. But the nightmare is that every single person showed up wearing the exact same metallic purple coat.
That is actually a brilliant analogy.
Thanks. So today we're zooming in on one specific attendee, Cortinarius alboviolaceus.
Also known as the Silvery Violet Cort, or the pearly webcap.
Right. We're going to figure out how to identify it, how to survive identifying it, because confusing it with its cousin can actually destroy your organs.
Yeah, the stakes are insanely high.
And ultimately, we're going to look at how the very concept of a species is being completely rewritten by modern genetics.
Which is a transition mycology desperately needed. The morphological disguise of these organisms is just too perfect.
So before we get into the DNA stuff, let's build that physical profile. Let's say you spot this thing in the woods. The cap is the first thing you see.
Right, the pileus. You're looking at something ranging from 2 to 9 centimeters across.
Okay, so fairly small to medium-sized.
Yeah, and when it first pushes up through the soil, it's shaped almost like a tight little bell. Or a hemisphere.
And it has a bump on top.
Yeah, a broad bump right in the dead center. Mycologists call that an umbo.
An umbo. Got it. But it doesn't stay bell-shaped.
No. As the fungal tissues expand through turgor pressure, literally pumping water into the cells to inflate itself, it flattens out.
Sometimes even turning its edges upward. Right,
Correct.
like a shallow helmet.
Exactly. But honestly, the shape isn't the real giveaway. It's the texture.
Because it looks like brushed metal. Which is so weird for a mushroom.
It is. The surface texture is what draws you in, and it's persistently dry.
Meaning no slime.
Right. Zero gelatinous slime, which is actually a really key differentiator from other Cortinarius mushrooms that are incredibly sticky or viscous.
Okay, so it's totally dry. But why does it look metallic? Nature rarely does something just to look pretty,
That's a great point. If you were to take a microscopic cross-section of that cap, you'd see the outermost layer, the cuticle.
Which is called a cutis in this case.
Yes, and a cutis means the hypha, those microscopic filaments that make up the fungus, are arranged strictly parallel to the cap's surface.
So they're just lying completely flat.
Exactly, stacked on top of one another like tiny microscopic logs.
Oh, wow. So it's structural coloration. A bit like a butterfly wing, right?
Sure.
To trick of the light.
Precisely. Because it lacks that gelatinous slime, those parallel dry fibers catch and reflect the ambient light uniformly.
It creates an optical illusion of spun silk or polished silver.
Yeah, it's not just a chemical dye, it's actual physical architecture reflecting light.
That is so cool. But beneath that silver sheen, there is an actual color, right? But the literature says it's a total shapeshifter.
Oh, it really is. It undergoes what we call an ontogenetic shift.
Meaning it changes completely as it gets older.
Right. In its primordial stage, the pigment is a striking pale violet, or a soft lilac.
But it's kind of hidden at first, isn't it?
It is. When it's just a tiny button underground, it's encased in a protective membrane called a universal veil.
And when it bursts through, pieces of that veil get left behind on the cap.
Exactly. That's what gives it that initial silvery white frosty coating over the lilac.
Okay, so it enters the stage looking gorgeous, but let's say a week passes, it gets sunny, it dries out. That lilac doesn't stay, does it?
Not at all. The pigment is super volatile. It degrades easily from UV light and just general environmental weathering.
So it basically bleaches out.
Yeah, it transitions into dull yellowish or grayish-white tints, usually starting from that center umbo and spreading outward.
And if the air is really dry, the cap literally starts tearing itself apart, right?
Yeah, the cellular structure of the cap margin contracts. It splits radially, creating this ragged, starry pattern.
So your beautiful metallic purple dome suddenly looks like a dried-out yellowing umbrella.
Exactly. It's a massive trap for foragers.
And if the cap wasn't confusing enough, the gills underneath are doing their own crazy wardrobe change.
Oh, the gills are doing a lot of work. They start out pale grayish-brown with just a hint of lilac.
But their whole job is to make spores. And spore production completely overrides the mushroom's color.
Right, because Cortinarius spores are a very specific, deeply pigmented, rusty brown.
And we're talking millions of them.
Millions upon millions. So as they mature, the sheer volume of rusty brown spores completely masks that original lilac color.
The gills go from lilac to cinnamon, and then to this dark clay brown.
Yeah, the spore factory takes over.
Let's talk about the spores themselves for a second. They're described as almond-shaped, but also varicose, meaning covered in warts.
They are very warty, yes.
Which seems counterintuitive to me. Wouldn't a smooth spore glide on the wind much better?
You would think so, but microscopic aerodynamics behave very differently than, macro aerodynamics.
How so?
Well, at that tiny scale, air is essentially a viscous fluid. It's thick.
Oh, interesting.
So those warts actually create a microturbulent boundary layer of air around the spore.
Like the dimples on a golf ball.
Kind of, yeah. That turbulence helps the spore stay lofted in the slight air currents of the forest floor, so it doesn't just drop straight down.
That is brilliant.
Plus, that textured surface acts like microscopic Velcro. It helps the spore stick to the precise microenvironment it needs to germinate.
Instead of just bouncing off a smooth leaf. Okay, that makes perfect sense.
Yeah, evolution is pretty clever.
So we've covered the cap and the gills, but we haven't touched on the name of the genus itself. Cortinarius comes from the Latin word cortina.
Right, which translates to curtain. It refers to the partial veil.
So most people know the veil on a grocery store mushroom, like an agaricus. It breaks and leaves a solid little skirt around the stem.
Yes, a membranous ring. But in Cortinarius, the veil is entirely different.
It's basically a cobweb, right?
Exactly. It's a fragile, intricate, spider-web-like structure made of fine silken threads stretching from the cap margin to the stem.
Hence the common name, webcap.
Precisely. But as the mushroom cap pumps full of water and expands, that delicate web has to stretch, and eventually it snaps.
So it collapses against the stem.
Right, it leaves a very faint fibrous zone. But remember those millions of rusty brown spores we just talked about?
The ones falling from the gills.
Yeah. Gravity pulls them down, and they get caught in the sticky threads of that collapsed cobweb on the stem.
Oh, so the stem basically acts like a catcher's mitt for its own spores.
That's a great way to put it. To the naked eye, it looks like a distinct rusty orange or brown dusting, forming a ring around the top of the stem.
That seems like an incredibly important field marker.
It is. And below that rusty ring, the lower half of the stem is usually somewhat club-shaped and sheathed in white, silky fibers.
From the universal veil.
Exactly.
Okay, let's just pause and view this holistically, because this explains why field mycology is notoriously difficult.
It is a nightmare.
Think about the timeline. It enters the forest as a pale lilac, metallic dome with a white cobweb. But a few days later, it's bleached yellow, the cap is split, the web has collapsed, and it's covered in rusty brown dust.
Yeah. If you find it on Tuesday, it's a purple jewel. If you find it on Sunday, it's a ragged brown mess.
So how on earth did early naturalists keep track of an organism that constantly changes its own identity?
Mostly. They struggled immensely, and they frequently got it wrong.
I bet.
The history of morphological taxonomy is just littered with errors because they were relying on temporal snapshots.
They were out there with magnifying glasses trying to count spore warts.
And trying to catch a fleeting glimpse of a cobweb before a rainstorm washed it away. It was almost impossible.
Which sets up the real danger here.
Yeah.
Because it changes color, it is practically begging to be confused with other purple mushrooms.
And there are a lot of other actors wearing very similar purple coats.
Right. Confusing this mushroom isn't just an academic oops. It is a potentially lethal mistake. So let's talk about the imposters.
We have to.
Let's start with its most dramatic cousin, the bruising webcap.
Ah! Cortinarius purpurescens. This is a critical lookalike. You'll find it in similar habitats, coniferous and mixed woodlands.
Especially around oaks and beech trees.
Yeah, and visually, from a distance, it strikes a very similar profile. A cap that's 3 to 10 centimeters, ranging from violet purple to reddish brown.
But the texture is slightly different. The Silvery Violet Cort is perfectly dry.
Right, but the bruising webcap has a slight tendency toward viscidity. In damp weather, it can feel somewhat sticky.
But the real defining trait is entirely chemical, isn't it?
Yes. This is where you have to transition from just looking at it to actively interacting with it. You have to damage the tissue.
You literally have to break it.
Yeah. If you take a bruising webcap and you score the cap with your fingernail, or crush the gills, the damaged flesh rapidly oxidizes.
It turns this vibrant, deep, reddish purple.
Instantly.
And the Silvery Violet Cort completely lacks this parlor trick. If I cut it in half, the flesh stays whitish lilac.
It absolutely will not flood with purple ink.
So, what is the actual biochemistry happening when I press my thumb into those gills? Why does a mushroom bruise? It doesn't have blood.
No, it's a brilliant localized defense mechanism driven by enzymatic oxidation.
Okay, walk me through that.
Inside the intact cells of the bruising webcap, there are enzymes, specifically phenyloxidases.
Like tyrosinase.
Exactly. And there are also separate complex organic molecules called polyphenol substrates.
And they're kept apart.
Yes. Normally, they are segregated in different microscopic compartments, usually vacuoles.
So it's literally like two liquid components inside a glow stick.
That is an excellent analogy. As long as the cell is healthy, they don't mix. But when you crush the tissue...
You break the compartments.
Right. The enzymes mix with the substrates in the presence of oxygen. The enzymes rapidly strip electrons from the substrates, converting them into highly reactive compounds called ketones.
And ketones are dark.
Well, the ketones quickly polymerize. They link together into long chains to form dark-colored, melanin-like pigments.
And that sudden dark pigment is the bruise. That is incredibly fast biochemistry. But why does the mushroom do it, just to warn us?
Nature didn't evolve this for human foragers. Those ketones are actually toxic and unpalatable to insects and slugs.
Oh, so when a slug takes a bite...
It instantly triggers this chemical reaction right in its mouth. It creates a nasty chemical barrier that stops the slug from eating more.
It's an immediate, localized chemical counterattack. That is so cool.
It really is.
But enzymes aren't the only chemical factories at work here. We have to talk about volatile organic compounds, or VOCs.
Yes. Because if the bruise test doesn't give away the imposter, the sniff test certainly will.
Because some of these purple lookalikes announce their presence with absolute olfactory violence.
Olfaction is a non-negotiable tool in field mycology.
Let's talk about Cortinarius camphoridatus, the goat-scented webcap.
Visually, in its lilac phase, it's a dead ringer for our target species. But the moment you bring it close to your face...
The descriptions of this smell are wild, universally horrific.
It hits you with a deeply foul, pungent odor. The most common descriptors are rotting potatoes, sweaty earth, or very explicitly, dirty socks.
Dirty socks, which points to a very specific chemical, right? Like butyric acid.
You're spot on. The sweaty odor is characteristic of short-chain fatty acids like isovaleric acid and butyric acid.
Which is the exact same stuff produced by bacteria on human feet.
Or the bacteria fermenting pungent cheeses. The fungus synthesizes these exact same compounds.
It is crazy that a fungus and foot bacteria arrived at the exact same smell.
Convergent chemistry.
Right. And then on the complete opposite end of the spectrum, we have the gassy webcap, Cortinarius triganus.
Another master of visual deception. Lilac on the outside, dry texture.
But if you crack it open, the flesh inside is saffron yellow to rusty brown, not whitish lilac.
And again, the smell is the immediate identifier. It doesn't smell like feet. It emits a heavily sweet, intensely fruity odor.
Like overripe pears, which sounds nice until it becomes nauseating. What's the chemical basis for the pear smell?
That artificial fruit aroma is almost always the result of ester compounds, like isoamyl acetate.
And why would a fungus want to smell like pears?
Well, while the dirty sock smell deters mammals, the sweet ester profile of Trigonus might actively attract certain flies or beetles to help disperse its spores.
So it's highly specific chemical communication.
Exactly.
Okay, so we have the dirty sock mushroom and the overripe pear mushroom. Both are purple webcaps. Both should be avoided.
Definitely.
But there's one final lookalike we absolutely must discuss. Because this isn't an imposter you want to avoid. This is one people actively hunt for.
Ah, the wood bluet. Lepista nuda.
Right, and making a mistake here is where people get seriously hurt.
The wood bluet is a choice edible mushroom. It's delicious.
Yeah.
And when it's young, it boasts a vibrant, beautiful purple coloration across its cap, gills, and stem.
So if you're an amateur and someone told you purple mushrooms in the fall are bluids, you're walking into a potentially lethal trap if you stumble into a patch of Cortinarius.
You really are. They look so similar at a glance.
So how do we deconstruct the wood bluet? What are the foolproof differences?
There are three non-negotiable differences. First, the veil. The wood bluet belongs to an entirely different family. It completely lacks a cortina.
So no silk and cobweb threads ever.
Never. Consequently, there is never a rusty brown, spore-dusted ring on the stem.
Visual check number 1. No cobwebs. What's number 2?
The absolute gold standard of fungal identification, the spore print.
Okay, how do we do that?
You take the cap, place it gills down on paper, cover it with a bowl for a few hours. The Cortinarius genus always produces rusty brown spores.
And the bluet.
The spore print of a true Lepistanuta is a pale pinkish buff, or light salmon. It is never ever rusty brown.
That's a massive divergence. Pink versus rust.
The spores tell the true identity, regardless of what color the cap is that day.
And the third check brings us back to the sniff test.
Right. The wood bluet doesn't smell like dirt or sweaty socks. It has a bright, sweet scent.
The foraging communities say it smells exactly like frozen orange juice concentrate.
Which is incredibly specific and accurate. It's likely a mix of terpenes and limon compounds.
Okay, so to synthesize. Purple mushroom. Smells like dirty socks. Leave it. Smells like overripe pears. Leave it. Scratches purple. Leave it.
Exactly.
But if it has no cobweb, drops pink spores, and smells like orange juice, you might have a bluet.
You might.
But this raises a terrifying question. Let's say I mess up. I ignore the rusty spores, I cook a true Cortinarius alboviolaceus in butter, and I eat it. Is this specific mushroom going to kill me?
This is where we discuss the unforgiving stakes of this genus. In any authoritative text, the edibility of alboviolaceus is listed as unknown, suspect, or strictly inedible.
Unknown is such a frustrating word in science. Does that mean we haven't found a toxin, or we haven't looked hard enough?
It means clinical toxicology hasn't isolated a massive concentration of a known lethal compound, specifically within alboviolaceus, to definitively classify it as deadly on its own.
But the risk is still considered absolute.
Because of its genetic neighborhood, it shares the exact same habitats and visual similarities with the most lethally toxic mushrooms on planet Earth.
Which brings us to the phantom killer, the orellanine controversy.
Yes.
What exactly is orellanine? I want to understand the chemical mechanics of why it's so feared.
Orellanine is a nephrotoxin. It specifically targets and destroys kidney tissue.
And biochemically.
It's classified as a bipyridine compound. If you want a chilling comparison, its molecular structure is strikingly similar to synthetic commercial herbicides like paraquat and dequat.
Wait, paraquat? The chemical used to completely defoliate weeds by destroying their cells.
Yes.
How does a mushroom naturally synthesize something so closely related to an industrial herbicide?
It's a terrifying evolutionary adaptation.
Now, what does a bipyridine molecule actually do when it enters a human kidney?
It initiates a catastrophic cycle of cellular destruction. When you ingest it, it absorbs into the bloodstream and filters into the kidneys.
Specifically the renal tubular epithelial cells, right?
Yeah.
The ones that filter waste.
Exactly. Once inside those cells, the bipyridine structure allows it to undergo what is called redox cycling.
Let me see if I can break down redox cycling. It means the molecule acts like a microscopic thief.
That's a good way to look at it.
It steals an electron from the cell's natural metabolic processes, turning itself into a highly reactive, unstable radical.
Correct.
But it doesn't stay that way. It immediately passes that stolen electron off to an oxygen molecule, creating a reactive oxygen species, like a superoxide radical.
Yes, and the terrifying part is
Passing that electron off resets the orellanine molecule back to its original state.
precisely. So it can immediately steal another electron and do it all over again.
It's a continuous loop.
It is a catalytic cycle of destruction. A single molecule of orellanine can generate a massive flood of reactive oxygen species.
Which overwhelms the cell's natural defenses, like glutathione.
And those superoxide radicals physically shred the cell's lipid membranes. They destroy its DNA. They halt the synthesis of proteins.
So the kidney cells literally suffocate, rupture, and die.
Yes. But the most insidious aspect of orellanine isn't just the destruction. It's the timeline.
Right. Because it doesn't happen immediately, does it?
The timeline is what makes it uniquely dangerous. It possesses an incredibly protracted latent period.
So if I eat a toxic webcap, My body doesn't trigger an immediate alarm.
No, you won't experience the rapid vomiting or GI distress associated with most poisonous mushrooms.
Because the orolini is just quietly setting up shop in the kidneys, starting that slow redox cycle.
Exactly. The oxidative stress takes time to accumulate. A victim might feel absolutely fine for 24 to 48 hours.
That's scary.
In many documented cases, the latency period stretches from 3 days to over 2 weeks before the first symptoms manifest.
A 2-week fuse on a biological time bomb.
Yes.
You eat it, you go about your life, you completely forget what you foraged. All while this invisible reaction is slaughtering your blood filtration system.
And by the time the symptoms hit the agonizing lower back pain, the extreme thirst, the metallic taste in your mouth,
What can doctors actually do for you at that point?
very little regarding the toxin itself. By then, the renal necrosis is advanced. The kidney injury is often irreversible.
So it's just supportive care, dialysis or a transplant.
Exactly. There is no known chemical antidote to orolini poisoning.
Which makes distinguishing these species literally an act of survival. But here's the paradox.
Okay.
If these mushrooms are visually deceptive, and eating the wrong one means delayed organ failure, how do mycologists safely study them? You can't just taste test a new purple Cortinarius.
Historically, they relied on animal models, which was slow and ethically complex. But modern mycologists have a macrochemical weapon.
Hoyland's reagent.
Yes, they can detect orolini without needing a multi-million-dollar mass spectrometer in the woods.
I've seen videos of this test. It looked like dark magic. It's a solution of iron further chloride, right?
FCl3, yes.
What is a chemical handshake happening when you drop that onto mushroom tissue?
It's a rapid demonstration of coordination chemistry. The bipyridine ring of orolini acts as a chelating ligand. When you apply the iron chloride, the iron ions immediately bind to the nitrogen atoms within the orolini's rings.
They lock together.
Tightly, forming an iron-orolinian coordination complex. And this new complex aggressively absorbs light in a very specific way.
Resulting in a color change.
Dramatic and instantaneous. The pale tissue suddenly blooms into an intense, inky, blackish blue or violet purple.
So a drop of amber liquid turns pitch black and you know you're holding kidney poison.
It's a vital diagnostic tool, but it underscores the golden rule. If you can't definitively differentiate the rusty spores and the cobweb from an edible bluet, you have no business foraging it.
The penalty for error is just too severe.
Way too severe.
This feels like the perfect pivot point.
Yeah.
We have a genus of thousands of species. They change color, they look identical, some smell like potatoes, some bruise purple, some contain a latent nephrotoxin.
What a mess.
By the late 20th century, the scientific community must have just thrown their hands up. Morphology had completely failed them. The visual world was a lie.
It really was. And that realization triggered a taxonomic earthquake. They had to abandon the visual world and plunge into the cold, hard data of DNA.
Let's trace the fault lines of this earthquake. Who was the first person to try and label the Silvery Violet Cort?
The formal record begins in 1801 with a mycologist named Christian Hendrik Persoon. He named it Agaricus alboviolaceus.
Wait, agaricus? Like the white button mushroom in the grocery store?
Yeah, you have to understand 1801 mycology, it was barely in its infancy. No powerful microscopes, degraded specimens.
So they just dumped everything with gills into a mega genus called agaricus.
Pretty much. Has gills meant is agaricus.
So it took a while for someone to realize a white spored mushroom with a solid ring is fundamentally different from a rusty spored mushroom with a cobweb.
Right. In 1838, Elias Magnus Fries, the father of modern taxonomy, recognized the difference. He moved it into his newly minted genus, Cortinarius.
Okay, so Fries puts it in the right genus, but Cortinarius is huge, thousands of species. How did they subdivide it?
For over a century, the logic was entirely textural. You group them by what they feel like.
Okay, so slimy ones together, scaly ones together.
Exactly. And because alboviolaceus has a dry, silky cap, it was put into a subgenus called Sericiocybe.
The silky heads, which makes intuitive sense. Yeah. If they both have silky caps, they must be siblings.
It was logical based on the tools they had, but it was profoundly wrong. When DNA sequencing arrived, specifically IE ISAR coding, they realized Sericiocybe was an illusion.
An illusion, like the grouping didn't biologically exist.
It was a polyphyletic grouping.
Oh, polyphily is such a mind-bending concept. It means the organisms don't share a recent common ancestor.
Right. They're completely disparate twigs from opposite sides of the family tree that somehow ended up looking identical.
Like carcinization in crabs. Evolution keeps inventing the crab shape from different ancestors because it just works.
Perfect parallel. Different lineages independently evolved a dry, silky cap. It's convergent evolution.
Because maybe the silkiness helped retain moisture or reflect sunlight.
Exactly. The environment exerted selective pressure. Grouping them by silkiness was like grouping bats, birds, and bees together just because they all have wings.
It fundamentally breaks the visual logic of the world. For hundreds of years, they organized a library based entirely on the color of the book covers.
Yes.
All the blue books in one room. And DNA finally let them open the covers, and they realized a romance novel and a physics textbook were shelved together just because they were both bound in blue leather.
That is the exact reality of the taxonomic earthquake. DNA allowed us to read the text.
So they evicted the Silvery Violet Cort from the silky subgenus. Where did it go?
It was relocated into a massive subgenus called Telemonia, specifically section Fermiores.
But reorganizing a 200-year-old library must be a nightmare. You can't just sequence a mushroom today and assume it's the same species Fries was looking at in 1838, right?
You've hit on the most agonizing bottleneck in taxonomy. It requires painstaking type studies.
What does that entail?
Cold case forensic science. Researchers travel to museums, request the original holotypic specimens dried by Fries and Pursoon centuries ago.
And they try to extract DNA from a 200-year-old crumbling piece of fungus?
Yes. The DNA is highly fragmented. They use advanced PCR to amplify the tiny fragments and piece together the ITS region.
And when they succeed?
They establish a neotype, the new definitive genetic barcode tied to the historical name. They plant a flag and say, this is the true Cortinarius alboviolaceus.
Okay, so the flag is planted, the European gold standard DNA is set, mystery solved, right?
You would hope.
Yeah.
But science is messy. When they compared that European DNA to silvery violet quartz growing in North America, the system broke down again.
Enter the North American cryptic complex.
Yes.
Let's talk about the 2025 CURO Symposium study out of the University of Georgia. They looked at mushrooms in Black Mountain, North Carolina.
Right. Led by Anna Satori Kinberg. They went out hunting for the pearly webcap.
And they found 12 mushrooms that looked exactly right. Metallic silver-lilac cap, dry texture, rusty spores, morphologically flawless.
Perfect matches to the naked eye.
So they take these 12 identical mushrooms back to the lab and extract the DNA. What happened?
They used ITS nanopore sequencing, and the results were antithetical to what they saw in the field. Those 12 identical mushrooms were genetically confirmed to be 9 entirely separate species.
I'm going to push back on this because my brain rejects it. If I hold 12 identical apples and a machine tells me 9 of them are oranges, I assume the machine is broken.
That is exactly the skepticism a good scientist should have.
How do we know it's not just natural genetic variation within one species? Like how humans have different DNA, but we're all Homo sapiens.
It comes down to what part of the DNA we're sequencing. We aren't looking at the whole genome, we're looking at the ITS region.
The internal transcribe spacer.
Right. It's a highly conserved region of ribosomal DNA. It does not vary significantly between individuals of the same species.
So it's like a license plate system that only changes when the actual make and model of the car changes.
Exactly. If the sequences differ by more than about 2.5 to 3%, they are reproductively isolated. They cannot interbreed.
And the divergence in those 12 mushrooms exceeded that.
Far exceeded it. The machine wasn't broken. It was revealing a truth our eyes couldn't see.
Okay, assuming the data is ironclad, how scattered were these 9 species? Were they at least close cousins in that Fermiores section?
That's the craziest part. They were scattered wildly. Some belonged to the section anomaly, identifying as Cortinarius iodes.
Wait, iodes? The Violet Violet Cort. Its defining trait is that it's incredibly slimy. How did a slimy mushroom end up in a collection of dry, silky lookalikes?
It perfectly illustrates how treacherous morphology is. Perhaps that specimen of iodes had been exposed to a prolonged drought.
So the slime completely dried up, mimicking the silky texture of alboviolaceus.
Exactly. The environment forced a disguise onto it, but its DNA betrayed its true identity.
Wow. Where did the others fall?
Across Telemonia, some in section Athabasci, some in Pseudotrigoni, and some actually in Fermiores, like Cortinarius subliquus.
But there's a detail in this study even wilder than misidentification. 3 of the taxa were completely undescribed by science.
Right. They didn't match any known sequence in any database.
A ghost species.
Completely unnamed ghost taxa hiding in plain sight.
This breaks my understanding of nature. I can hold a silver mushroom in North Carolina and an identical one in Sweden. Flawless copies. But genetically, completely separate entities.
That is what we call a cryptic species complex. Genetically isolated but morphologically indistinguishable.
But why? Why don't they evolve to look different?
Because of stabilizing selection. The environment dictates the rules for survival. In those specific microclimates, a dry, silvery lilac umbrella is the absolute mathematically optimal tool for thriving.
The environment is carving the exact same statue out of completely different blocks of genetic marble.
It is a profound realization. Our eyes are only registering a superficial fraction of the reality. The true genetic reality is infinitely more complex.
Which naturally leads us to the ultimate question. What are they actually doing down there?
They are performing a critical job.
Right. They aren't just sitting there confusing mycologists. They are running the forest. Let's talk about the wood wide web.
Yes. The profound ecology of Cortinarius.
Now, wood wide web is catchy, but it glosses over the brutal transactional reality. Fungi are heterotrophs. They can't photosynthesize, they have to eat.
Right, they can't make sugars from sunlight.
So how does an organism that can't make its own food survive underground?
By forging an intimate partnership, nearly all Cortinarius species are obligate ectomycorrhizal fungi.
Ecto meaning outside, myco for fungus, rhizal for root.
Precisely. The mushroom above ground is just the apple on the tree. The actual body is the mycelium, a vast network of microscopic threads called hyphae in the soil.
And these hyphae seek out the rootlets of living trees.
When they make contact, they wrap around the root, forming a physical sheath called a mantle.
Like a microscopic glove over the tree's roots.
But they also grow inward, penetrating the spaces between the root cells to form a complex labyrinth called the Hardig net.
But they don't breach the cell walls.
No. The Hardig net just creates a massive shared surface area between the fungus and the tree. This is the interface, the trading floor.
And what's being traded on this microscopic stock market?
The fungus acts as an external stomach and mining operation. Tree roots are blunt. They can't access tiny pores in the soil.
But the microscopic fungal hyphae can.
Exactly. They secrete enzymes that dissolve rock, mining for distant minerals, nitrogen, phosphorus, zinc, and they pump these materials, along with water, directly to the Hardig net.
And what does the fungus demand in return?
Carbon. The tree is photosynthesizing, making liquid sugars. It pumps up to 30% of those sugars down into its roots across the Hardig net to feed the fungus.
Carbon for phosphorus, sugars for water. But the Silvery Violet Cort seems to adjust its portfolio depending on the continent.
Regional host switching. It's fascinating. In Europe, the true alboviolaceus partners strictly with broadleaf hardwoods like birch and beech.
It ignores conifers.
Entirely. But across the Atlantic to the Pacific Northwest.
The rules invert.
Completely. Those alboviolaceus-like cryptic cousins partner exclusively with giant conifers, Douglas firs, spruce, hemlock, pines.
That adaptability is staggering.
Yeah.
But I have to ask, how do we actually prove this trade is happening? We can't see sugar flowing into a hypha underground.
It's not just a theory. Scientists prove it using stable isotope tracking.
By tagging the atoms.
Exactly.
Right.
Elements like carbon and nitrogen exist in slightly different isotopes, like carbon-13 or nitrogen-15, which are heavier.
So they introduce heavy carbon to the tree canopy and heavy nitrogen to the distant soil.
Yes. Like putting microscopic heavy GPS trackers on the molecules. They wait, then harvest the mushroom cap and run it through a mass spectrometer.
And the mass spectrometer vaporizes the tissue, ionizes it, and shoots it through a magnetic field.
Right. And because carbon-13 is heavier, its momentum makes it take a wider turn through the field, hitting a different part of the detector.
So it literally sorts the atoms by mass.
And by reading those signatures, scientists definitively prove that the heavy nitrogen in the mushroom came from the soil meters away, and the heavy carbon came from the tree canopy days prior.
It transforms the wood wide web into measurable infrastructure, which means that single purple mushroom is actually an interface terminal for a massive network connecting dozens of giant trees.
It's an indicator of ecological antiquity. You only see these massive Cortinarius networks in ancient, undisturbed forests. Without them, those giant trees simply couldn't survive. The trees are the architecture, but the fungi are the lifeblood.
It completely shatters the illusion of individuality in the forest. Trees are just nodes on a fungal grid.
They really are.
Let's take a breath and synthesize all of this. We started with a visual anomaly, a silvery lilac cap. We decoded its structure, its structural color, its warty spores, its collapsing cobweb.
We navigated the biochemical forensics, the bruising quinones, the dirty sock volatile compounds, and the terrifying redox cycling of orellanine.
And then we plunged into the genetic revolution. from Elias fries sorting by texture to modern nanopore sequencing, proving that a single disguise hides 9 different ghost species in North Carolina alone.
All acting as the invisible mining infrastructure of the forest.
It is a staggering amount of hidden complexity. But before we wrap up, I want to leave you with a disruptive thought.
Oh, Marty.
We spent an hour talking about how DNA lets us draw hard lines around what a species is. But think about those cryptic species in North Carolina.
Okay.
If an entire complex of genetically distinct organisms has independently evolved to look exactly the same, down to the spore warts, and they perform the exact same ecological function, is our human obsession with drawing taxonomic lines missing the point?
It is the ultimate philosophical question of biology. We love our neat, labeled boxes.
Exactly. But nature doesn't read our textbooks. Perhaps in fluid, interconnected, underground world of mycelium, the concept of a species is just an arbitrary human illusion that the fungal kingdom has already outgrown.
Or perhaps it's a boundary they never truly fit into in the first place.
Thank you so much for joining us on this exploration into the astonishing, deceptive world of the pearly webcap. Keep your eyes on the forest floor, and always remember to question the reality of what you are seeing, because beneath every fallen leaf there's always a deeper mystery waiting to be unraveled.